Chapter 28 is about the nervous system. So we've talked about the fact that there are two systems that kind of regulate or are in charge of all the other things happening in the body. One of those is the endocrine system, and we've looked at that with looking at endocrine glands and some hormones, and then even in the reproduction chapter, we looked at hormone control. And so now we're going to look at the nervous system. So this is the brain and neurons. So this picture, these green entities on your picture are neurons, nerve cells. So they have a cell body that contains organelles just like every other cell, but then they have these long extensions off of them. On one end, there's a very long, a little bit thicker extension called the axon, and at the other end, there are extensions called dendrites. So we're going to learn more about the exact structure of the neuron and how neurons work together to send signals around the body. So this, again, goes over the idea of these two systems that are responsible for being in control of the functions of the animal body, the endocrine system and the nervous system. So we see in this slide that the nervous system can be broken down into two parts, first of all, and that's indicated at the bottom of the slide. The central nervous system and the peripheral nervous system. The central nervous system is in the center, that's the brain and the spinal cord, and all the nerves coming off of the spinal cord compose what we call the peripheral nervous system. So let's look at how we perceive things. First of all, there has to be some kind of input, likely from the environment, that's picked up by the brain. So here we have an insect biting us or stinging us, and we pick up that signal, and neurons are going to send that signal to the brain. The brain's going to figure out what to do, and it's going to send something back. In this case, you're going to swat at that area or grab it with your hand. The next slide is going to go over this in words. So we break down the entire nervous system into two parts, the central nervous system and the peripheral nervous system, right? You saw that in the last slide. And the central nervous system consists of the brain and the spinal cord. And the peripheral nervous system consists of nerves. And nerves are made of bundles of neurons. So neurons are nerve cells. But you put lots of these together to form something called a nerve. And those neurons are wrapped together in connective tissue to form what we call a nerve. There also are ganglia, G-A-N-G-L-I-A, which are clusters of neuron cell bodies. So we see those in the peripheral nervous system as well. This slide shows you the continual breakdown and layout of the system, with the peripheral nervous system being divided into two parts. The sensory part, which is going to get signals from our sense organs and send them to the spinal cord and to the brain for processing. And then the other part of the peripheral nervous system is the motor division, which is going to conduct signals from the brain back out to different parts of the body. And you'll see that that motor division divides up again into different systems. And we're really, again, just going to scratch the surface of the nervous system and really trying to understand how nerve signals move around the body. So this is a nerve cell or neuron. And if you look, you can see the enlarged part, the cell body, which is where the nucleus is, and all kinds of other organelles are in there as well. And then you have these extensions. Shorter, thinner extensions along one end of that cell body, and the other end a long extension called the axon. If you look at the scanning electron micrograph, you will see the cell body is very large compared to these extensions. The extensions help nerves send signals very quickly. If nerve cells didn't have these long extensions, you'd be relaying messages from one cell body to another cell body to another one. It would take much longer than these long extensions that can reach out and make these connections much more quickly. So we see a couple other things here. There's extensions coming off the cell body that receive signals are called dendrites. The part of the neuron that's going to send a signal to another neuron or to some other cell, that's the axon. The axon, you'll see, has some coatings on it, the layers of myelin, and that helps insulate. Because let's think about these signals. These are really electrical charges. And just like you have along your wires in the house, you have insulation. Think about a cord to a lamp, for example. It's not just wires, it's surrounded in some insulation to keep the electrons flowing along that instead of, leaving the wire totally. So that's exactly what's happening here. We have those myelin sheath, that myelin sheath. In between, some neurons, in between places of the myelin where there's no myelin, there are what's called nodes of Ranvier, and they help signals travel even faster. They kind of jump. If you see there, that signal pathway, jump from node to node. So sometimes that will happen in some neurons. At the end of the axon, we have the endings, the axon endings, which are also called the synaptic terminals. And they're going to be releasing things that may be picked up by a cell or by the next neuron. So based on what I've already said about these neurons, the highly branched short extensions are, of course, the dendrites, D-E-N-D-R-I-T-E-S, dendrites. And then the axons are longer. So take a moment to really think about the structure of this nerve cell. Why does it look the way it does? Why does it have these long extensions? How quickly do these signals need to go from place to place? Well, think about how long it takes you to decide to move your finger or to say something or anything or blink your eye. These we think are instantaneous, but they take time, but just a tiny, tiny fraction of time because neurons are very good at relaying signals quickly. So now we're going to delve into how the signal is propagated from neuron to neuron, how the signal moves from one neuron to the next. So we're going to talk about when the neuron isn't doing anything, when it's just resting, it's not sending a signal first. And at rest, the neuron's plasma membrane has a membrane potential called the resting potential, which results from the positive charges on the outer membrane surface and negative charges inside. The resting potential exists because of ion concentrations forming a gradient across the plasma membrane of a neuron. You'll see that in the next slide. So here's a neuron at rest. And if you see that up close part of the cell, the axon part of the cell, you will see on the inside of the axon, the inside of the cell is slightly negatively charged, and on the outside is positively charged. That's what happens when the cell is resting. That's the normal situation for neurons. So if you also look at that up close and personal, at the bottom, we have all kinds of things here we need to sort out. So first of all, we need to find the plasma membrane, and we have some proteins stuck in there, and they are channels. They help things move. We know that there are transport proteins in the cell membrane. So here's one that's a sodium channel that's going to move sodium. We also have a potassium channel that's going to move potassium. And then we have another one that you've learned about a couple times, and certainly in Bio 101, the sodium-potassium pump, a transport protein that moves things in two directions. Sodium one way and potassium the other way. Sodium-potassium pump requires ATP to function, whereas the other protein channels do not. They are passive. They're relying on a concentration gradient to move things, right, from high to low. You're going to need to watch this animation a few times. It's a lot of information in it. So be listening for the idea of the resting potential, the fact that when the neuron isn't doing anything, there is potential energy stored across the cell membrane. So make sure you understand that. And then make sure you're following how exactly this charge difference across the membrane is maintained. So the neuron gets a signal, okay? It gets a signal, and that is going to cause a change in the membrane potential. So think about the resting potential, and now that's going to change because the stimulus is going to alter the permeability of a portion of the membrane, allowing ions to pass through and changing the membrane's voltage. Okay. A nerve signal, also called an action potential, is a massive change in the membrane voltage from the resting potential to a maximum level and then back again. Think of it like a wave of energy moving down the axon, or a wave of electricity moving down the axon of the neuron. The image here is important to understand, so you'll read this several times before you actually grasp what it's saying. But of course, start with number one. Number one is the resting state, which you've already looked at, where there's positive outside the membrane and negative inside the cell, inside the neuron. And then you'll see that you go to depolarization, where it's going to open some channels. And we're going to start moving ions, and that's going to start changing the charge across the membrane. Then more channels open, and we have this propagation of a signal, and then the channels close. And then we have to reset back to our resting state. So go through this, read it carefully, look at the pictures, and make sure you understand what's happening on the cellular level, the cell membrane, as the action potential is moving down the cell membrane. Look at what's happening to the different transport proteins. Which ones are opening? What ions are moving? How is that affecting the charge? The previous few slides went over the action potential and how it moves along the axon. So here we're kind of just putting some ideas on top of what you just looked at. An action potential is a localized event. There shouldn't be a blank there, sorry. It's a localized event. Self-propagated in a one-way chain reaction along an axon, and it's an all or nothing event. You either have an action potential or you don't. You don't have different levels of action potential. So what happens is the strength of the stimulus, how the nervous system perceives that differently, is not in the strength of the action potential, because the strength of the action potential is always the same, but it's the frequency of repeated action potentials. Another image showing you the same idea of an action potential moving down the cell membrane of the axon of the neuron. And what is happening to the sodium ions and the potassium ions. So this is repeat information from the previous slides. Just the image is a little bit different. This short video will put things into motion for you. So you've looked at a bunch of images. You've read and looked at how some of those transport proteins open, those channels open, and things move across the membrane. So now this is going to go over that same information, but now it's going to be in motion. So this might be a little bit clearer to you. So what happens when this electric charge gets to the end of the axon? What happens when it's run down the whole axon by this action potential at different localities along that axon, and now you've propagated an action potential? What happens when it gets to the end, when it gets to the axon endings? Okay, well, it's going to probably want to signal the next neuron in line, and it does that by producing a molecule, sending a signal across a very short distance. That distance, that space, is called the synapse. The transmission of signals between neurons or between neurons and effector cells. So an effector cell could be a muscle cell or just any other kind of body cell that's being affected by the neuron. So the transmission of signals between the neurons or between neurons and effector cells occurs at junctions called synapses. There are two types. At electrical synapses, electrical signals pass directly between the cells. At chemical synapses, the sending neuron secretes a chemical signal. That signal is called a neurotransmitter. And that tiny molecule will cross the synapse, cross what sometimes is called the synaptic cleft. It's the space between neurons, and will bind to a receptor on the surface of the receiving neuron, the receiving cell. Pictured here is the end of one axon at the top. So that's called the sending neuron. So that's one of those little axon endings up close. And so you'll see how there are going to be some molecules that are going to leave a vesicle that's going to have these neurotransmitters. The cell makes them, right? It makes all kinds of proteins, and the cell can make neurotransmitters, packages them in a vesicle. The vesicle is going to merge with the plasma membrane at these tips, at the axon endings. And these neurotransmitters are going to go into the space in the synapse called the synaptic cleft. They'll be picked up on the other end. So at the bottom, you have a dendrite. It's the receiving neuron. This is the end of a dendrite. And the dendrite is going to have in its cell membrane some receptors, right? Some proteins that function as receptors. And they're going to pick up the neurotransmitter, and that's going to cause something to happen. So if you look at that picture very carefully, you'll see that the proteins, these transport proteins that have the neurotransmitter attached to them, are open, and ions are moving into the cell. You'll also notice over on the right what happens once the signal is done. Once you've passed it from neuron to neuron, you want the signal to end, okay? And so what happens is sometimes the cell will take back up the neurotransmitters and use them at another time. Or sometimes they're broken down by enzyme action in the synaptic cleft. So those neurotransmitters don't stay around for a very long time. This little animation will put things in motion for you. So that last image, it'll put this in motion and go over exactly what happens at the synapse. This slide is a kind of a dose of reality. Just when you think you understand how one neuron is sending information to another neuron and how you might get a signal from your brain to somewhere else in the body, we throw this in here and say, well, yeah, that's what happens, but it's actually much more complicated. Some neurons excite the receiving cell and allow an action potential to form. Some stop it. And a cell doesn't just receive one signal. It can receive signals from many neurons. So this can become quite complicated. There are lots of different kinds of neurotransmitters, and they're used across that chemical synapse, so produced by a neuron and move across the synapse to send some kind of signal to the next neuron. The one you are responsible for knowing is the neurotransmitter called acetylcholine. It is released by motor neurons. Motor neurons are neurons that are going to activate skeletal muscles. So think about as you move through the day, as you raise your hand, as you sit in your chair, you're sending signals from your brain to all these muscles. And the neurons ultimately are signaling the muscles with this chemical, right, with this little chemical signal called acetylcholine. Other neurotransmitters that you might have heard of are norepinephrine, serotonin, and dopamine. So norepinephrine is an excitatory neurotransmitter in the peripheral nervous system, and it can also function as a hormone. So sometimes it goes from neuron to neuron, and other times it's picked up by the circulatory system and moved somewhere else, like another hormone, like hormones. Serotonin and dopamine are released in the central nervous system, and they affect sleep, mood, attention, and learning. So these are some other neurotransmitters you might have heard of before. This image looks similar to one a few slides ago where we looked at the synapse, but this one's a little bit more specific. This one's talking about a neuron that's going to signal a muscle cell, okay, down at the bottom. And so here it's very specific what we're looking at. We're looking at the action potential reaching the axon ending and causing the synaptic vesicle to release not just any old neurotransmitter, but acetylcholine, because we want to signal a muscle cell. And so acetylcholine is a very specific molecule, and it will have a very specific receptor on the muscle cells. So again, you'll release the neurotransmitter. It will go to the muscle cells, bind to the receptor, and it will activate the contraction of the muscle. That acetylcholine in that space that isn't picked up has to be broken down. And it is rapidly hydrolyzed, right, by acetylcholinesterase, by an enzyme in that synaptic cleft, and broken down into acetate and choline. And then the neuron, the signaling neuron, right, the axon, is going to absorb that back up and make more acetylcholine. So a little bit of recycling taking place there. A little bit different view of what happens between the neuron and the muscle cells. So we see our end of the nerve cell, so end of an axon ending there. We have that neuromuscular junction, or the synapse, the space between the neuron and the muscle cell. So neuromuscular junction. We have the acetylcholine, there's little black triangles being released and being picked up by receptors on the surface of the muscle cell. So what happens when that happens? So great, so you have acetylcholine attaches the receptor. What does that cause to happen inside the muscle cell that makes the muscle cell contract, become smaller? Well, it triggers the release of calcium ions that were stored in the endoplasmic reticulum of the muscle cell. So in the muscle cell, the endoplasmic reticulum has a very specific name, sarcoplasmic reticulum. And that calcium that is released causes the cell to contract. So if there's a problem with the nervous system, if it's not working properly, and we want to treat that, a great place to treat it would be at these synapses in between neurons. And this is where many drugs act. And when I say the word drugs, I don't mean just, illegal drugs. I mean all these drugs, prescription drugs, over-the-counter drugs, even things that we don't even really think of as drugs, like caffeine, nicotine, alcohol. These can all affect the action of neurotransmitters in the brain synapses. The image shows an example here of a neuron releasing the neurotransmitter serotonin, and then it being able to reabsorb serotonin. And then if you put a drug in there, the drug can work on those receptors and inhibit the uptake of the serotonin from that synaptic cleft. So if you look in that diagram on the right, the SSRIs, serotonin inhibition, okay, is taking place because those receptors are not letting serotonin bind and be taken back up by that neuron. Let's step back from what's happening, microscopically at each neuron and between neurons to send signals around the body, and look at the nervous system as a whole. So animals have a very similar nervous system. So we have the hydra picture there, and we have the salamander. And in both cases, there are lots of nerves, lots of neurons, rather, lots of nerve cells. Through the course of evolution, we see an organization of the nervous system. We see the organization into something that we now call the brain, centralizing where information goes and where reactions come from. So radially symmetric animals, right, like the hydra, have a nerve net. Bilaterally symmetric animals show cephalization, sort of the idea that there's a head. Cephalization, the concentration of the nervous system in the head end of the organism. We have centralization, the presence of a central nervous system, including the brain and the nerve cords. Ultimately becomes the brain and the spinal cord in complex animals. Here's a good thinking question. So let's say it's a snake. Why is it advantageous for all the snake's sensory organs and its brain to be at the anterior end, at the head end of the organism? Think about that. We talked about the central nervous system and that the brain is the master control center, and that the spinal cord is a bundle of nervous tissue or nerve cells running lengthwise down the spine. So the spine or the spinal cord is not one neuron and one neuron, it's bundles of neurons, bundles of nervous tissue. So this diagram shows again the difference between the central nervous system and the peripheral nervous system. The brain and the spinal cord obviously connect. If you look at the brain image there, the lower part, you'll see that you have the spinal cord where it attaches to the brain. So it's going to get signals directly from those neurons in the brain and be able to send them down the body. Over to the right, you have a cross section of the spinal cord. Gray matter and white matter, that's just the orientation of how the neurons sit in the spinal column. And coming off of that bundle of nerve tissue, you have other nerves, spinal nerves, part of the peripheral nervous system, right? Because they're going to take signals not just up and down the back, but outward from that spinal cord to the arms, down the legs, to vital organs. The peripheral nervous system is broken into two parts. The motor system has to do with motoring or moving, so that's the skeletal muscles. And then the autonomic nervous system, which regulates and sends signals to all kinds of other things that we don't even think about all the time. Controlling the heart, controlling smooth muscles, moving food through the digestive system. Sometimes sending signals to endocrine systems so that certain hormones are released. When we think of the autonomic nervous system, autonomic, think of it as automatic. So this is involuntary. In our next chapter, we'll talk more about these sensory neurons, how we get information from the environment, and then we grab that through our sensory organs, through our sight, through our taste, through our hearing, and send signals to the brain, and then the brain processes, interprets that, and then sends signals back. And again, just a breakdown of those systems. And this is showing you that autonomic nervous system broken down into parasympathetic and sympathetic and enteric. And in general, you don't have to remember or know anything about that. We'll keep things at the level of the autonomic system, just the things that happen involuntarily that the brain has control over. And here we have the peripheral nervous system and just a recap of what we've already talked about. So looking at the brain, just looking at some of the different parts of the brain because they do different things. In the course of vertebrate evolution, the forebrain and the hindbrain became more subdivided into specific regions in more complex animals. The cerebrum's outer region, the cerebral cortex, is really important in highly complex more recently involved organisms like mammals, like humans. The cerebral cortex is responsible for a lot of thinking and learning that we associate with the higher order of organisms. So as the brain develops in the embryo, generally we see three main regions occur: forebrain, midbrain, hindbrain. And then as development continues, we have more specialization. The forebrain divides in the cerebrum and other parts of the brain listed there, the thalamus, the hypothalamus. We've talked about that with our endocrine system, right? Posterior pituitary, pineal gland, two endocrine glands from the forebrain. And then the hindbrain divides up into some other parts of the brain that you can see there. And that happens very early on. On the right is the fetus at 3 months old, and we've already laid down the basic brain structures. The brain is an amazing organ. So it's got over 100 billion neurons, but it's also got other cells. It has other cells that support those neurons, that help them function and give them the nutrients they need for the important job they're doing. So the brain is highly complex. It's only been in the last 20 years that we really are starting to understand exactly how the brain functions. And a lot of that has to do with the ability to image the brain and to see what's happening in the brain while people are looking at things or moving. So you're going to see the midbrain and subdivisions of the hindbrain and the thalamus and hypothalamus are really important in conducting information to and from the higher brain centers in the cerebrum. And what does the brain do? It regulates all kinds of things, that midbrain and hindbrain. Regulates homeostatic functions, all kinds of levels in the body, keeps track of your body position, sorts out all this sensory information coming through your senses into the brain all the time. Here we have the major structures of the brain. And the cerebrum is the largest and most complex part. And it gives us a lot of skills and functions that we think are really unique to humans. And we know that we have memory and learning and speech and emotions. And sometimes we don't know if other animals have the same abilities that we have, because we have a very highly convoluted cerebrum and a very large brain. This image just shows you all those brain parts we talked about in the previous slide. This is the image if we were standing behind somebody and looking at their brain. So the right side, the left side, the medulla oblongata there, the cerebellum towards the back. The picture here is from the side of the brain, so orient yourself. And here we have where they've mapped the brain and what parts of the cerebral cortex are important in different functions. So you see we have the sensory cortex, the motor cortex, visual, auditory, So it shows where sensory information is received in the brain. This is how we know so much about how the brain works from this imaging. And that's our overview of the brain and more in-depth understanding of how nerve signals move around the body through the neurons from one neuron to the next. And these are your objectives. So please, as you're going through the slides and reading the book, make sure you are writing these objectives. And when you write these objectives, it's kind of the more work you put into them, the more you're learning as you go, and the better you'll do on exams. If you just write a few words, if you just scratch the surface of what the objective's actually asking you to do, then you don't have complete answers and you don't have a complete understanding. So spend some time and develop these responses, properly and thoroughly.